If you are weak or have forgotten the basic fundamentals of acid, bases, and pH, please use the PowerPoint presentation that is included under the tutorials for Chapter 26 before starting this 26 Part 2 tutorial. Where do we gain access to hydrogen? Well, a little bit is in our diet through the consumption of fatty acids and amino acids. We also metabolically form some acids. Remember that if we combine carbon dioxide and water, we get carbonic acid. We can also metabolically produce lactic acid and also keto acids. Our normal plasma pH in your textbook says 7.35 to 7.45. This slide is from the other text where the range is a little more narrow. I would accept either. Now, to maintain that pH, we have three mechanisms. We have our chemical buffers, we have our respiratory system, and we have the renal system. Our buffers and the respiratory system do not eliminate hydrogen ions from the body. They just convert them into a different form. Our renal system is the only way we can eliminate hydrogen ions from the body. Let's take a look at these three mechanisms. Before we can talk about the body's chemical buffer systems, I want to make sure you're clear on what a buffer does. Adding a buffer to a solution that has a low pH will bring that pH closer to 7. Conversely, if you add a buffer to a solution that has a high pH, it also will bring that solution closer to 7. In other words, with buffers, we don't have wide swings of pH, too low or too high. Let's look at some examples here. For example, if you have a strong base, you can buffer it with a weak acid. And this chemical reaction between the two will form a weak base and water. So the weak base has a pH that's closer to 7 compared to the strong base. An example is one weak acid we already know is carbonic acid. A strong base that doesn't exist in our body is sodium hydroxide. If we add carbonic acid to sodium hydroxide, it forms water and a weak base, sodium bicarbonate. Sodium bicarbonate is one of these chemicals that can act either as a salt or a weak base. So take a look at that tutorial, the PowerPoint that I added, if you're confused about that. Well, the same trend exists if we need to buffer a strong acid. To buffer a strong acid, we could add a weak base. When we mix those two together, the chemical reaction results in a product that is a weak acid and another product that is a salt. So, for example, we know a weak base, sodium bicarbonate, and a strong acid, hydrochloric acid. When they are mixed together, the chemical reaction will proceed to form a weak acid, carbonic acid, and a salt. When looking at our chemical buffer systems, again, we can use these various chemicals listed here to contribute either a weak acid to buffer a strong base or a weak base to buffer strong acids. Our bicarbonate buffer system is really important in terms of extracellular fluid buffering. It is the main one. We may also see it in the intracellular fluid. Phosphate buffer systems is important in terms of the buffering the urine and the intracellular fluid. And then our protein buffer systems. This doesn't happen all the time, but we have some proteins that have amino acids on one end that have a carboxyl group, and the other end have the amine group. And the carboxyl group can act as a weak acid, and the amine group can act as a weak base. In other words, the carboxyl group can donate a hydrogen, and the amine group can take up a hydrogen. The protein buffer system is very important in the intracellular fluid. We also see it function some in the blood plasma. And then earlier in the semester, we saw the protein called hemoglobin function as a buffer. As I've mentioned, the most important chemical buffer in the body is the bicarbonate ion. Now, we've seen this formula before. We have seen it in the respiratory system. This is one I told you to tattoo on your forehead. This is a reversible reaction. It happens in our blood plasma all the time, and it reverses all the time. Why would it reverse? Well, first let's talk about what's normal, and then we'll talk about changes. So the normal ratio of carbon dioxide to bicarbonate is 1 to 20. So for every 1 carbon dioxide, there will be 20 bicarbonate ions. Let's take a look at this formula relative to changes in carbon dioxide and or bicarbonate. So first, let's take a look at what happens when carbon dioxide increases. In other words, instead of 1, let's make it 2 molecules of carbon dioxide. And at first, before equilibrium is reached, there will be 20 molecules or ions of bicarbonate. What happens? Well, that is not chemical equilibrium. That is not the normal ratio. And so we say that as carbon dioxide increases, the formula is driven to the right, and that will result in an increase in bicarbonate. By the way, it also increases hydrogen, which can lower the pH. So once equilibrium is reached, instead of 20, it should be 40 ions of bicarbonate. Now, what happens if the bicarbonate increases? Remember, our normal is 20 ions of bicarbonate for each molecule of carbon dioxide. Well, if we increase this to 40 ions of bicarbonate, then the formula is driven to the left. In other words, this bicarbonate ends up picking up that hydrogen, and therefore the result is a decrease in hydrogen and an increase in pH. So when equilibrium is reached, then, if we started with 40 bicarbs, we will end up with 2 carbon dioxide at equilibrium. If we have bicarbonate in the plasma, it will pick up free hydrogen. Therefore, it is called a buffer, and that's why we say the bicarbonate buffer system is so dominant. That is our main chemical buffer system. What other mechanisms are available in the body to buffer and prevent swings in pH? Well, your respiratory system can contribute to maintaining pH. Here we see the same slide I showed you in the respiratory chapter. We know that as CO2 increases in the cells, it will move down the partial pressure gradient into the plasma. When it goes into the plasma, when I say it's dissolved, that means CO2 plus H2O yields what? You remember? Carbonic acid, which can dissociate into a hydrogen ion and a bicarbonate ion. So in the plasma itself, carbon dioxide can contribute bicarbonate ions. But how does it contribute the most? We know that about 70% of all carbon dioxide is transported in the plasma as bicarbonate. Boom. We have our buffer there. Just one thing to point out here before I mention what is going to happen if the reverse occurs. There is an example of a protein acting as a buffer. Hemoglobin picking up a free hydrogen. If you recall, there are receptors, both central and peripheral receptors, for changes in carbon dioxide, hydrogen concentration, and to a lesser extent, oxygen. And those receptors are what will increase or decrease the respiratory rate. If you increase the amount of carbon dioxide that is being transported in the plasma, and that plasma goes past the medulla oblongata, it will be detected and you will have an increase in respiratory rate. When you have an increase in respiratory rate, the result is you blow off more CO2, and therefore CO2 levels will drop back to normal. This is how the respiratory system can influence pH. We'll look at this in more detail in class. Let's look at the flip side of that. What if you have a decrease in carbon dioxide in the plasma? What effect does that have on the respiratory rate? Do you remember? it will also decrease. And therefore, with a decrease in the respiratory rate, you will blow off less carbon dioxide, you will conserve carbon dioxide, and therefore the carbon dioxide comes back up in that case. Again, we'll look at these mechanisms more closely in class. Although the chemical buffer system and the respiratory system can alter the pH of the blood, neither one of them can eliminate hydrogen ions from the body. They just convert them from one form to another. It is only through the renal physiology that hydrogen ions can be eliminated from the body. Let's take a look at that, looking at this slide. Our first step here is to recognize that this is going on in the proximal convoluted tubule. There are other mechanisms in the distal convoluted tubule that we'll talk about next. But let's start here, recognizing that the proximal convoluted tubule can help eliminate hydrogen ions. The first step here is carbon dioxide that is picked up from the filtrate can combine with intracellular fluid water in the presence of carbonic anhydrase to form our carbonic acid. Now, I know it seems silly, but it has to happen. The next step is to dissociate that acid into a hydrogen ion and a bicarbonate ion. The bicarbonate ion ends up being transported out of this tubule cell into the interstitial fluid here and subsequently into the peritubular capillary. Most of that happens thanks to secondary active transport mechanisms on the basolateral side that are driven by the sodium concentration in the cell and the chloride concentration in the plasma and the interstitial fluid. Bicarbonate ions are moved out of this cell and back into the plasma. What happens to this hydrogen ion? Well, hydrogen is secreted into the lumen, into the filtrate. For every hydrogen that is secreted, a bicarb is moved into the peritubular capillary. Now, this is important in terms of understanding how the kidney can get rid of hydrogen ions. If a patient is experiencing acidosis, that means their pH has decreased. Remember the normal range, about 7.35, this is your textbook, to 7.45. I've seen narrower ranges, like 7.38 to 7.42, but this range will do. So in acidosis, a patient's pH is at 7.35 or lower. That means this patient has too many hydrogen ions. This is one way that the kidney can eliminate hydrogen ions. I'm using this illustration to show you how else hydrogen can be eliminated from the body. This is from the other textbook. I just think it's more clear than the one in your textbook. There are unique cells found in the collecting duct that are called intercalated cells. We have two types, type A and type B, and what you're seeing here is type A. Type A intercalated cells are functioning if a patient is in acidosis. If they are in acidosis, that means the concentration of hydrogen is high. Bicarbonate functions as a buffer. This will form carbon dioxide and water. The carbon dioxide diffuses into the cell. It joins with the water inside the cell in the presence of carbonic anhydrase, and it dissociates back into the hydrogen ion and the bicarbonate ion. If you'll recall, hydrogen ions do not diffuse readily across most membranes, so that's the only way we can take it from the interstitial space and get rid of it in the filtrate and eventually in the urine. We have to convert it into a form that then can be absorbed by or have it transported into this cell. Now, once this step has happened, we again have a hydrogen ion and a bicarbonate ion. These intercalated cells are interesting. In the type A cell, we see our old friend, the bicarbonate chloride antiporter. We saw that in the membrane of the red blood cell. This works the same way. And so this cell can put the bicarbonate back into the interstitial fluid to act as a buffer for more of this hydrogen. What happens to the hydrogen? Two things, both of which require ATP because we're moving the hydrogen against the concentration gradient. One mechanism is to use a hydrogen-potassium antiporter. Potassium is moved into the cell when hydrogen is pumped out of the cell. The other mechanism is just a hydrogen pump, sometimes called a proton pump, where ATP again is used to pump hydrogen from inside the cell into the filtrate. Eventually, those hydrogens will be excreted in the urine. Now, there is only one little problem, potential problem here. When a patient is in acidosis and the renal tubule is getting rid of the hydrogen, it is in exchange for potassium, and more potassium is reabsorbed. It is possible that this patient now will have a problem with hyperkalemia. We'll talk about that in class. Before we move to the next slide, I just want to point out on the basolateral side, we have the antiporter that handles bicarb and chloride. And on the apical or luminal side of these cells, we have the ATP-driven hydrogen exchanger and pump. Now, let's move to the next cell. Take a look at this cell. Is it the same cell we saw in the previous slide? Well, it has some of the same transporters. Here's the bicarbonate chloride antiporter. And here are the ATPase driven pumps. But if you notice, they're on the opposite sides of the cells that we looked at in the previous slide. These are type B intercalated cells. And essentially, they work in reverse as the type A. And so these are the cells that are going to go to work if a patient is in alkalosis. If a patient is in alkalosis, they have too few hydrogen ions. Their pH is too high. And so if you'll take a look at these cells, you will see that hydrogen is conserved and bicarbonate is secreted and eliminated. We see a possible problem also here relative to the potassium. When we conserve hydrogen, reabsorbing hydrogen, we will secrete more potassium. And it is possible that the potassium concentration in the body goes too low, and that is called hypokalemia. Again, we'll discuss that in class. This is a really good flow chart that shows you, again, we have, because of the law of mass action, we have a relationship between hydrogen concentration and partial pressure of carbon dioxide. We talked about that in chapter 22. We talked about how the peripheral chemoreceptors as well as the central chemoreceptors can influence the respiratory center. If plasma concentration is too high, that means the pH is low, and so this patient is in acidosis and needs to get rid of hydrogen. So how does that happen? Well, the respiratory control center gets more messages saying, okay, let's, through the phrenic nerve, increase our respiratory rate. It also increases the depth of breathing. You end up blowing off more carbon dioxide. That lowers plasma partial pressure of carbon dioxide, which in turn lowers plasma concentration of hydrogen, which means the pH is brought back up to normal. This is the respiratory mechanism. Can we look at this slide in reverse? What if you have a patient that is in alkalosis? pH is too high. What does that mean? Well, plasma hydrogen is low. Plasma PCO2 is low. Again, both the peripheral and the central chemoreceptors respond to that. How do they respond? Well, fewer signals reach the respiratory centers in the medulla. This will decrease the output from the medulla. So the action potentials in the phrenic nerve decrease. That suppresses the muscles of ventilation. The result is a decrease in respiratory rate and depth of breathing. In other words, in this situation, we are conserving carbon dioxide instead of exhaling it. What is the result? Well, plasma CO2 would increase, causing plasma hydrogen concentration to increase, and that would result in a decrease in pH. Again, this would be for a patient that is in alkalosis. This mechanism returns the pH back to the normal range. Why is it so important to maintain the pH within that narrow parameter? Well, we know that changes in pH can denature proteins. This can be very problematic if you think about all the various proteins within the body. We have transport proteins on the cell membrane. We have hormones that are proteins. We have hemoglobin that is a protein. The other effect of pH imbalances that we could see are changes in the respiratory rate. We could see changes in potassium balance, either hyper or hypokalemia, and ultimately those things can lead to cardiac malfunction. When you first look at this slide, you say, oh, this is the pH scale. But in fact, it's a very narrow piece of the pH scale. Remember, 7 is neutral. But our plasma is slightly alkaline, and the normal range is between 7.38 and 7.42, or in your textbook, 7.35 to 7.45. This is from the other textbook. And so Dr. Silverthorne's range is more narrow. Below 7.35, a patient is in acidosis. Compensation is possible. In other words, the body's mechanisms can come into play to correct acidosis, renal and respiratory system. It may not fully correct the problem, but there will be an attempt at it. Same thing with alkalosis. When the pH is higher than normal, there are mechanisms that the body uses to compensate, to try to bring the plasma pH back within the normal range. Sometimes it gets to normal, sometimes it just approaches normal. Let's take a look now at these acid-base disturbances. Before we get into detail on the pH imbalances, I want to let you know that what I am teaching you about this is what I call my down and dirty, simple way of looking at pH problems. Doctors, emergency room personnel that take a look at these cases that have disturbances in pH and likely disturbances in electrolytes, it's not just a science, it's an art. The more experienced the doctor is, the more likely they will understand how to apply fluids that can correct imbalances. My way is not that sophisticated. It's not just my way. I think when you start nursing school, you will also see something very similar. So we're going to keep it simple. We have 4 categories of pH imbalances. Respiratory acidosis. Why respiratory? That's what causes it. In respiratory acidosis, the pH is dropping because the respiration rate is too low. By conserving too much carbon dioxide, that also increases hydrogen concentration, decreasing pH. But the respiratory system is not the major cause of acidosis. Acidosis can also be caused by metabolic problems, either within cells in general, or it's possible the kidney is not working correctly. Either way, there is an increase in hydrogen ions and a decrease in pH. Respiratory alkalosis is caused by a problem in the respiratory system. If somebody is hyperventilating, blowing off more CO2, the hydrogen concentration decreases, causing an increase in pH, respiratory alkalosis. Metabolic alkalosis, the pH is rising, hydrogen concentration is dropping. Again, this can be caused by cells in general or problems with the kidney. This is a table from your textbook, and it's not easily read. So my next slide takes this information that you see over here and puts it into the table that I hope will help you understand it. One thing to add to your table on the next page is your knowledge about causes that is given to you here. This is a very simple way to take a look at acidosis and alkalosis problems. Across the top here, we have the condition, and you can see the four that we've already talked about. We have what kind of shift in pH would occur. We take a look at PCO2 values and also bicarbonate values. Now, bicarbonate, as most electrolytes are, or ions are, is measured in milliequivalents per liter. You don't need to understand that measurement. You will need to memorize all of the values that are across the top here. All right, let's take a look at what causes these problems. Taking a look at respiratory problems. If the respiratory rate decreases, there will be an increase in PCO2. And that will result in a decrease in the pH. In other words, increased PCO2 increases hydrogen concentration, which decreases the pH. Now, if there's compensation both through your chemical buffers and the kidney, there will be an effort to make and conserve more bicarbonate. And so this value will increase. The opposite occurs in respiratory alkalosis, often caused by hyperventilation. If you are breathing out more CO2, your PCO2 drops. If PCO2 drops, hydrogen concentration drops, which means pH increases. If the various mechanisms are effectively compensating, they will be conserving hydrogen ions and dumping bicarbonate ions. So the bicarb levels will drop. Next, let's take a look at metabolic issues. Now, in metabolically caused problems, we will have these shifts that you see here, but the respiratory system will attempt to compensate. So we know that if pH drops, that's because hydrogen has increased. That will increase rate of respiration. The body blows off more carbon dioxide, and so PCO2 will drop. The other thing is bicarbonate levels in the plasma drop because it is attempting to buffer the hydrogen ions. In metabolic alkalosis, we have too few hydrogen ions and the pH increases. The respiratory system will attempt to compensate. It will slow respiration, conserving carbon dioxide, and so PCO2 will increase. In addition, bicarbonate levels will increase. I have two final points to make. One is, how are you going to remember this information in an easy way? Well, there's no easy way. You just have to memorize these values. But to remember what condition is what, I want you to notice the difference in the direction of the arrows here. In conditions that are caused by respiratory changes, the arrows are not all the same. They point in different directions. You can see it here in respiratory alkalosis also. But if the condition is caused by a metabolic problem, all three arrows point in the same direction. Again, in metabolic alkalosis, all three are pointing up. So that is a way to almost cheat, if you will. It's not cheating, it's using a memory technique to be able to remember what's what. One more point. If the problem is caused by the respiratory system, by hyperventilation or hypoventilation, it cannot be the system that compensates. If it is caused by respiratory problems, the respiratory system cannot compensate. Also, here in terms of metabolic alkalosis, if the cause of the metabolic alkalosis is kidney failure, or metabolic acidosis is kidney failure, the kidneys cannot compensate. They are the cause. So try to keep that straight also. The cause cannot be the compensation.